I think we'll get started. Yeah, the introduction. My name is Jun Li. I'm a faculty in Department of Human Genetics and also Computational Medicine and Bioinformatics. It's my pleasure to host today's seminar organized by the Single Cell Spatial Analysis Program. A few housekeeping items. I believe we have started recording for this seminar.
That is correct.
Yeah, I kind of want to quickly thank Vanessa Evans, who is the program manager, who did all the behind-the-scenes work organizing this, along with Sunita McGrath, who is a co-director of one of the leaders of the program. I want to remind everyone that our next seminar is not 1 month from now, but moved ahead to December 6th. It's also a Monday afternoon at 1 p.m. from Dr. Rahili Rabari, who's visiting virtually from the Wellcome Sanger Institute, hosted by Tom Wilson. So I hope you can attend. I believe her talk will be more on the single-cell somatic lineage delineation and somatic genome evolution. All right, so now turn to today's speaker. We thank you, Dr. Nils Gehlenborg, for visiting us. Dr. Gehlenborg is currently a faculty member in the Department of Biomedical Informatics at Harvard Medical School. Years ago, he received his Ph.D. from University of Cambridge as a pre-doctoral fellow at the European Bioinformatics Institute. His research is on developing computational techniques and interfaces for biomedical data to enhance the capability for visualization so that he can support reproducible collaborative research. He has been very active in developing data portal and user interfaces for various publicly funded, highly visible consortium efforts and applying these tools to cancer genomics, epigenomics, and chromosome conformational studies, and so on and so forth. He is also active in his service to the community. He's co-founder and former general chair of BioVis Symposium on Biomedical Data Visualization and co-founder of a number of other entities, as you can see in the introductory paragraph in the event announcement. He also serves as a director of the Masters of Biomedical Informatics program in the Harvard Medical School. I had a chance to previously interact with Dr. Gehlenborg, mostly in his role as the key person organizing the data portal for the NIH HubMap project. And today his talk, as you can see, the title is Integrative Visualization of Multi-Modal and Spatially Resolved Single-Cell Data. So thank you, Nils. I think you can start.
Thank you so much for the very kind introduction, Jun, and thank you to all organizers for inviting me, and in particular, thank you to Vanessa for being very patient with me, with sending in my abstract and all that. So I'm really happy to see so many of you here today. And as Jun said, I'm going to talk about data visualization today. And before I get that started, I want to say one very important thing, right? So data visualization is something that is designed for humans, and humans play a very big role in my lab, not only in terms of the research that we do in data visualization, but also because I like to hire more humans into my lab. So I have multiple postdoctoral fellowship, postdoctoral positions open in my lab that are related to the work that I'll be presenting here today. So if you're working in the field of imaging, spatial omics, single cell analysis, and you have an interest in coming here to Boston, to Harvard Medical School and work with us, please reach out to me. You can either direct message me on Twitter or send me an email. I'm sure you'll find my email address, and I would really be delighted to hear from you. So the challenge that I want to talk about today that we're trying to address is very nicely summarized by the following overview slide here. So I'm as you just heard, I'm part of the HubMap consortium. HubMap is a program funded by the NIH Common Fund to essentially build an atlas of the human body at single cell resolution across a lot of different tissues. And as part of this effort, a lot of data is being generated. And I'm using this slide to motivate our work, because what you can see here is an overview of the assays that are being used, the technologies that are being used to generate data for the HubMap program. So as you can see here, the list is rather long. If you look at the y-axis on this plot, you know, you're ranging from sequencing assays to untargeted LCMS, which is a mass spectrometry assay. There are microscopy-based assays, more sequencing assays, various multiplexed immunofluorescence assays such as Codex, and then the list goes on. So it's an extremely diverse collection of technologies that people are using to generate data of a wide range of human tissues. In this case, the list is actually fairly short in this screenshot here from our data portal. If you go there today, you'll see more tissues, and in the future, you'll see even more tissues. So I think we're up to several dozen technologies and several, probably close to 24 different tissues. And so what is the problem with this, right? So if you're trying to create tools that allow people to view this kind of information, this kind of data, in a data portal, for example, but also, of course, you're not just looking at data in a data portal, you're also looking at data when you're analyzing it. And when you're maybe sitting there at 11:30 p.m. on a Sunday night in you know, in front of a Jupyter Lab notebook and trying to you know, generate some results that you can present on a Monday morning in the next lab meeting. So the challenge here that you will face is, right, when you have so many different data modalities, how do you bring all of this information together in a visual tool that allows you to interact with the data and explore the data? And then on top of that, there's another issue, and that is related to the scales, right? So as I mentioned, HubMap is meant to build an atlas of the human body. And so clearly when we're looking at the human body and then someone says single cell, so there is an enormous range of scales, right? So there you can look at an organ, but then you have organ parts, you have functional tissue units. So in a kidney, for example, you have glomeruli, which are made up of multiple cells, and then you have, of course, individual cells. And so the scales here are enormous. And then from a more technical point of view, apart from dealing with all the different data modalities, for us as developers and designers of data visualization tools, we also, of course, need to take into account that, you know, not everyone is going to use Python. Some people will be using R to analyze their data. So and then, you know, once you get into R or Python, then people will probably want to use their favorite package to do single cell analysis. Maybe, and in fact, it seems like everyone has their own favorite, and then they have one that's coming out of the lab, and usually it's the same. So we have a really, really rich ecosystems, which is great. But at the same time, as you're thinking about building tools that should be universally applicable, that needs to be taken into account. And so this is very specific to spatial. Well, these are challenges related to spatial and single cell data visualization. Now, when you're looking at you know, the tissue atlas projects, here are just a few, and there are many more. So I think there are probably dozens of tissue atlas projects at this point. These are some of the bigger ones that I'm somewhat more familiar with, such as HubMap, or then the NIH Human Tumor Atlas Network, HTAN, which is a sort of a sister project that's focused on cancer atlases, while HubMap is focused on healthy human tissues, or the Human Cell Atlas. And then, of course, many tissue-specific ones or organ-specific ones. KPMP is another example, the Kidney Precision Medicine Project, which has many people at the University of Michigan. I'm sure some of, I maybe I hope some of them are here, and their projects like gut map and lung map, et cetera, et cetera, et cetera. Right. So, and all of them are generating data using different modalities. And I think this is an issue that we're facing right now, is like, there are just many contenders, new technologies that people have developed in the last couple of years, and we're trying to figure out, which ones are going to be the ones that we're going to be using, sort of like, who knows, maybe there won't be any de facto standards, but I think over time, things will normalize a little bit, and there will be maybe a fewer number of technologies that people will be using to do these kind of studies. But right now, it's really, anyone can bring their own tool, and we'll see how things go. So often, I think there's also a challenge. And now I'm again talking sort of from the data or from the tool builder point of view. It's not always clear, what are all the different kinds of analyses people want to do. A lot of this is brand new, right? People haven't done projects at this scale yet. So if you're trying to build a tool to support certain types of analyses, it's often not really clear, what people ultimately want to try to accomplish with the tools that we're trying to build them. And here I'm really talking about visualization tools. And then when we're dealing with Atlas, in particular whole, body atlases, the amount of data that's being generated is huge, right? So this is not something that is easily managed. So we need to be able to access large sets of data, but we cannot simply download these data sets generally, right? So they're often even too big for individual submitters, and HubMap. There are several examples where people submit their data via FedEx, right? Still like shipping a hard drive to the data coordination center. And that is sort of the scale that we're dealing with. Okay, so let me talk a little bit more about the solution that we propose for this issue. And the solution is called Vitesse. I'm gonna really go right into it. Here's just the cover page of a preprint that we've posted. I don't think I included the URL. I'm happy to put this in the chat in a moment. And we describe a tool to visualize a multimodal, spatially resolved single cell data. But of course, this could be unimodal, it could be a single data modality. It also doesn't have to be spatially resolved single cell data. So as you'll see in a moment, this tool is rather flexible and can really do a lot of different things. And what I'm hopefully going to be able to do today is I will hopefully be able to convince you that what we're building here within the context of HubMap, but also now applying to other projects, is something useful and that you can maybe apply in your own research if you need visualization tools, and you will hopefully find this helpful and not a problem as you start playing around with it. So I'm sharing the URLs here for you right now. I know some of you might just be, oh, thank you, Vanessa, for sharing the link too. Some of you might be more interested in the actual software than listening to me. So let me tell you a little bit more though about the background. Vitesse is a tool that is web-based, and it can do a lot. So let me tell you a little bit more about how this fits into a bigger ecosystem of tools, and how it actually, and like what it can do. So let me just walk you through this tool ecosystem. And in this tool ecosystem, because of the spatial aspect of the data, imaging plays a central role. And so this is why we have, as one of the major components of this Vitesse tool, we have something called Viv. And Viv is a JavaScript library that we developed to visualize multi-scale, high-resolution, multiplex bioimaging data in a web browser. This is really, really important. There have been other tools in the past that people developed, and I will tell you towards the end of my overview here a little bit more about why we developed our own. But Viv is this JavaScript library that is, something you can apply to imaging data, and you can render it in a web browser very conveniently without having to have any special server infrastructure. That is something that we really focus on. We want to make it as easy as possible for anyone to put imaging data somewhere on a web server and then display it in a web browser using our tools. And related to Viv is sort of something called Avivator, which is a viewer for images that is built around this library and sort of shows off all the different features that this tool has. So Avivator looks like this. You can see there's an overview on the right with all your different channels. You can add channels, remove channels, set the color palette here and the color mapping. And you can see at the very top here, you can't see my mouse pointer, I presume, but you can probably see this. So here there's just a URL, and that's where the image sits, and that's all you need. So is this web server. And then what you can see here is a lens. So we have a flexible framework in Viv that allows you to write code that can be shown as a lens on top of the image. So you can modify the image for in a specific well-defined region. Okay, so this is Avivator, and whoops, and then moving on, if I can, yeah, here we go. So another tool that you might be interested in is Vizar. Vizar is a client for ZAR-based images. ZAR is a sort of emerging standard to represent multi-dimensional data sets and particular imaging data sets that comes out of the Python world. And Vizar is a tool that you can run or plug into a Jupyter notebook, as you can see here. Very, very simple. All you need to say is, what is your image? Where's the location of your data? And then you say, run this thing, and you get another image viewer here that can interact directly with the data in your notebook as well. So that's Vizar. For those of you who are familiar with the imaging standards, OMI-TIFF, or the OMI next generation file format, OMI NGFF, Avivator, Vizar, and Viv support all of these formats and are designed around imaging standards. Very, very important, because if there were no standards, we couldn't build tools to interact with that kind of information. All right, so enough about the imaging. So now I want to talk a little bit more about Vitesse. Vitesse incorporates this image viewer. And Vitesse is an integrative visualization tool. So you can not only look at images, but you can bring in all sorts of other data. And so this is one example of what Vitesse can look like. Vitesse can take many, many different forms. It is a multi-view tool, so you can have multiple views that you can link together for different types of interactions. You don't really see this in these screenshots. But here in this example, you have two views of this is a Visium data set, where on the right, you're showing expression levels for a selected gene, and on the right, on the right, we have expression levels, and then we have the same image on the left here at the top, where we show cell type assignments, right? And so you can see how the cell type assignments correlate, or maybe not correlate, with the expression levels of the selected gene in this case. So, in this implementation, in this example of the test, we've configured it so that when you zoom in or pan in these image views here at the top, they will both move together. But we could also set it up differently so that one of the views shows an overview and the other view would show you a detail. So you can set up an overview detail configuration. And you'll see many, many more examples here as we go along. And I just want to really point this out because Vitesse is really a framework that allows you to build applications for different problems in spatial single cell data analysis. All right, and then moving on, so Vitesse can be used as a standalone tool, but it can also be embedded as a component in a data portal. So this is the HubMap data portal with a view of Vitesse, in this case showing a codex data set. And you can see here we have a single spatial view. We have a different configuration of the application. So it can do that. It's a React component you can just embed in whatever application that you have. Here's another example where we've basically just taken Vitesse and we're showing it on its own website as a simple app. So you can point Vitesse to a configuration, and once again, it looks completely different. I mean, at this point, you probably recognize certain components, image components, or the spatial view, the heat map view. But we can also incorporate views like this one down here, where we can look at expression quantifications per cell type, or cell set is what we call these things. So cell sets are either a number of cells that you select in a view. It could be predefined cell type assignments, and then you can use these groupings of cells to, for example, display the expression levels of a selected gene of interest. That's down here in the bottom right is this view that I'm trying to highlight here. Oops, it's okay, it's gone. So these are examples of Vitesse as a standalone tool or embedded in another application, such as a data portal. But of course, in these settings, Vitesse is primarily a viewer for data. And clearly, if you just have a viewer for data, it comes with a number of limitations, right? So for example, you're looking at a data set, you look at a cluster in there or a cell type, and then you have additional questions about it. So you can't really answer this with Vitesse if it's embedded as a component somewhere. But where you could answer that is if you had access to the data and some analytical tools. So that's why we also support the integration into other analytical frameworks, such as the aforementioned Jupyter Notebooks. So here is a view of Vitesse embedded in Jupyter Lab. So it's an IPython widget that you can launch directly from your Python code. So all you need to do is you essentially create a configuration for the visualization that you want. In this case, we're looking at ATAC-seq data. You can see here's a full-blown genome browser in there. This is another tool that my lab developed a couple of years ago called HiGlass that can be used to display genome map data. And so even though this is static here, this is actually a browser that allows you to view the whole genome or individual genes or whatever you want to do. And you can see here we map the color of these ATAC-seq tracks to the clusters shown in the UMAP plot on the top right here. So we can do that. And of course, as I said before, some people like Python, other people like R. So we also have an HTML widget for those of you who are familiar with the R world, and that allows you to embed this here either directly in an RStudio session, or you can embed it in a Shiny app. And then you go to the website, you'll see demos and examples for all of those. So we have all of this available. And in this case, you could run a SERAP-based analysis and then visualize the results directly in Vitesse. In this case, we show two different algorithms and then a comparison of the cell type assignments generated by those. So that's why there are two scatter plots. They're linked together, so they will zoom and pan in this case. When you zoom into one, the other one will follow, and the panning is linked as well. So this is an overview of the tool ecosystem that we're providing. So there is Viv, the JavaScript library, and then derived viewers for the imaging side of things. And then there's Vitesse, which embeds Viv. And as I said, it's available as a JavaScript component or a React component. You can embed in a website, or you can build your own tool around it. And then there's the Python package and the R package. I should add that the Python package is fairly stable. The R package is a bit more experimental right now. But this is something that we're planning to address going forward and really create equal functionality for both of them. All right, so now in addition to the tool ecosystem, we also need to talk a little bit about data. As I mentioned before, Viv supports the OMI standard, and so we have support for OMI TIFF and then the OMI NGFF, which is a ZAR-based format. And so there's a preprint now that describes this paper. I think I have a slide. Oh yeah, it's coming up. So in that paper, you'll see that VizR is one of sort of the implementations that already support the OMI NGFF. And then OMI compatible ZAR is sort of an intermediate format that was in use for a while in the past. And then Vitesse itself is also using ZAR a lot, because ZAR has the big advantage that you can just put a ZAR store. ZAR store is essentially a directory structure of lots and lots of individual files. And you can put this either in an S3 bucket or other cloud storage or on a web server, and then access these things without the need for any other servers. And you can also access parts of a data set very, very conveniently in ZAR. And that's why it's so powerful. So Data, a very popular container for single cell data coming out of the scan package, is something that can also be stored in ZAR. And so we're making extensive use of that. We can read genome map data out of a ZAR-based format as well. And then we support the image formats. And we have a bunch of custom formats that we support as well that are usually in JSON, such as image segmentation masks, etc. But that's actually something I want to point out. Unfortunately, this is like the data formats and standardization is still early days. As the more analysis people do, the more results they produce, and often they don't really fit into the existing formats or containers, and then they come up with their own solution. And I think we're guilty of that as well. We have some custom formats for some of our data and that we can display in Vitesse. And I think here it's really a call to action to the community to get together and figure this out. And there are some ongoing efforts across these different Atlas projects to standardize on say feature by observation matrices, or otherwise known as cell by gene matrices, to extend these containers and describe a schema that allows people to share data across different tools and projects without having to constantly map data back and forth between different formats. Okay, so by talking about formats, just two examples here. So one on the left, there's this preprint describing the OMI NGFF paper. So Trevor Mance, who is a PhD student in my lab, who developed Viv as sort of the lead author, he's a co-author on this OMI NGFF paper, which is, I think it's going to come out in Nature Methods very soon. So this was led by the OMI community. And then another issue is not so much about what you how you store the actual imaging data, but another one is about the metadata. And so this MITI, the Minimum Information Guidelines for Highly Multiplexed Tissue Images, is a project led by Peter Sorger and others in the Human Tumor Atlas Network. And that is a really, really important issue as well, to have metadata available about the data that is stored in these images. So as tool builders, we really, really need these efforts. We need standardization, because otherwise it's hopeless to build something that is broadly applicable across different, analysis frameworks, et cetera. So we really try to get involved in these efforts too. And I'm very excited that many in the community are so good at collaborating and often sort of very altruistically spend their time on doing something that is not going to directly lead to their next Nature, Cell, or Science paper, but will make it easier for everyone in the field to make progress on data analysis and data sharing and data visualization, of course. So I want to point out how we are doing the rendering in the Viv image visualization library, because it makes such heavy use of these standards. I just want to show you really quickly how image rendering on the web was done prior to the Viv image rendering library. So there are two approaches, or it can distinguish two separate approaches, basically. And they can be called offline server-side image rendering or online server-side image rendering. And so let's look at offline server-side image rendering for a moment. So there's a primary data set, which in this case would be maybe an OMI TIFF file. And then in order to render such an image, there would be a pre-processing step, which has a data transformation in it. So like the OMI TIFF doesn't specify any color mappings or anything like that generally. You have just the different channels in there. And the color mapping and all that would be applied in this data transformation step. And then what this data transformation step does, or the offline image rendering step, it generates an image pyramid. Image pyramid's very important that for zooming and panning, right? So when you zoom into a very large image, you want to maybe start with an overview. And in order to display an overview, you don't want to transfer the whole image at the highest resolution. You want to transfer a small preview, essentially, and then look at the preview. And then as the user decides where they want to look in detail, you only transfer the data needed for the region that they're interested in. So that's why we store image pyramids, which consist of lots and lots of small image tiles, which in this case are served by a web server, and then you render this in the client. So this image tile is usually a PNG or a JPEG file. And there are a lot of different libraries out there that support that. OpenSeaDragon is one very popular example that a lot of people use to visualize these kind of image pyramids that were pre-generated essentially using this offline server-side image rendering approach. The alternative to that is the online server-side image rendering, where you have the image, you can retrieve data tiles from the image, and then you have a specialized server that will on demand essentially take this data tile and turn it into an image tile and send it over to the client, which will then render it. So similar idea, but the difference here is the timing when the data transformation happens, right? So in the online server-side image rendering, it's happening at the time when the server, when the client, the web browser requests a specific image. So you can change settings on the fly, right? You could change the colors on the fly very easily. You just need to tell your server, hey, I want this channel in red instead of blue. And then the server can do this. This is not so easy, or in fact, it's not possible to make such changes when you have a pre-generated image pyramid that was generated offline, where the client cannot communicate with the data transformation step. So you can see the pros and cons. The pro of this online server-side image rendering is clearly you can make changes quickly on the fly. However, the con here is you need to maintain a server, and you need to run a server, and a server needs resources. So if you have 1 million people accessing this thing, your server might become overloaded very quickly. Now, few people have scientific images that are being viewed by 1 million people, but nonetheless, there is a cost in setting this up and maintaining this infrastructure. The offline image rendering has the advantage that you essentially just need to put your image pyramid or your image tiles in a directory in a web server, and then it can be accessed with an appropriate image rendering library that knows about the structure, but you cannot make any changes to the color mapping or colors in general, right? So that's the downside. And so what we proposed is this approach by Viv, and what we not only proposed but also implemented is essentially a method to do the data transformation directly in the client, so in the web browser itself. So here, the data, the raw data, or well, the raw data stored that is in the primary data file, so an OMI-TIFF file, is transferred all the way to the client, which will then do the rendering on the fly. And so here, the advantage is we can change image settings very, very quickly. We can modify the colors, the color mapping, and anything else directly in the web client, which makes these things extremely responsive. And we don't have any need to maintain any server infrastructure in order to enable that. And so that's really the big advantage. So now we have a simple JavaScript library that anyone can use, and you just point it to your OMI-TIFF file or OMI-NGFF file, and you can get started. So that's what we developed here, and we were able to do this by harnessing or employing very modern web technologies. In fact, our image rendering approach is built into something called DeckGL, which is a rendering library that came out of Uber back in the day and was used originally for maps and other spatial data. And so we built our layers, our image rendering layers into that framework, and we're able to really build on some other community developed framework. And so this is now available to anyone who wants to use it. And I hope you will give it a try if you ever need to render any images. Okay. I want to move on and talk briefly about image segmentation. This might actually, I'll keep this fairly brief here. So although there are a couple of slides, I'll go through them quickly. So what is very important, of course, for anyone who's doing analysis of spatial data is the ability to identify regions of interest, such as cells or functional tissue units or anything like that. And so we are currently supporting 3 different data formats. So one is our custom format that we started out with, and we're supporting OMI-TIFF bitmasks. So in the OMI-TIFF bitmaps, the segmentation masks are stored in an image file. And then we supported QPath format, which is something that is built around GeoJSON, which is another mapping, originally developed for maps and mapping format that we can use. In fact, the coming soon can be deleted. We have it already rolled out. And, and then we can, of course, render or annotate whatever levels. So the segmented objects, right, they can be grouped together. So cells could be grouped to and assigned to a cell type. We can map expression levels or other quantifications to objects, such gene expression levels or anything else. And then you can, of course, no matter what approach you use, whether it's an OMI-TIFF bitmask or our own format or QPath format, you can select these objects or these entities and get more information about them. Right? So here's sort of an example with glomeruli that were segmented manually in a QPath actually, and then saved as a QPath file. And so you can see here's like, here's the segmentation. Nothing exciting in here for anyone who has dealt with this, but you can select these things in the test, and then you can, see expression levels or whatnot. Here's an example for an OMI-TIFF-based mask. So this is just the image, and here's now it's the segmentation. You can see we can have multiple objects at the same time. So there are cells, nuclei, and then cell and nuclei boundaries in this case. So here we're highlighting the nuclei and the cells, and then you can get the boundaries if you just care about the nucleus boundaries. Or in this case, I think there's one more in here, the nuclei and the cells as well. So, and here are only the cells. So that's all possible in Vitess, which means we can link these spatial views that start out as an image, but we can now link them to our cell level information, for example, which can be displayed in this heat map down here, or you have the cell sets, which is a way to represent all sorts of collections of cells. All right, and then going forward here, as I mentioned, linking this to gene expression level, so we can overlay, in this case, it's DAPI, which is not that exciting. Here's something else. This is CD45. You can see how this is all mapped on there. I think for anyone who has worked with an image viewer and dealt with imaging data, this is maybe not so surprising. But the advantage here is clearly this is running in a web browser. There's no need for you to run anything else. And in fact, Vitess also does not rely on a backend server. So you can, all you need is a web server that you need anyways if you have a website. But Vitess runs exclusively in the client, so in the web browser. There's no need to set up any infrastructure and maintain that infrastructure. Here's an aspect of the integration, right? So when I hover on a cell, I can see, in this case, there's not much to see in the heat map, but I can see the heat map where that cell is, and then I can look at various markers in the heat map as well. Okay. As I said, I want to move through this rather quickly. So one thing that we haven't quite resolved, but something that will happen and that we'll work on is 3D segmentation, right? So we can view 3D data as well in Vitess, or in this case, it's Avivator, which is our, again, this is Avivator is built around Viv. So the image rendering library, Viv, can not only do 2D images, it can also do 3D images. And again, it can be multiplexed and it can be multi-scale. So this works just beautifully. You can see this here. And again, there's no server. So this is all running in your web browser. And you can see, we can add additional channels to that. And in this case, it looks kind of cool. This little examples is 3D image mass cytometry data that you can view in here. But now, of course, this is just viewing the data. Like the next step would be to have a segmentation and then being able to turn on and off certain cell types and then just get the subset of the volume that corresponds to certain cells. So that is something that we are working on, and you will see this in the future, but certainly it's not been fully solved, at least not in the context of the test. There are certainly more advanced tools that are custom applications for tools to generate this kind of data where you can do this, but we don't have that yet in the test. Okay, so I want to show you a couple more things briefly here, and then sort of wrap this up. I have a hard time, of course, judging who's here. So I will briefly, well, I've already shared the link for you for the website. So we have the Vitess website, and this beta will go away any day now, but we'll make sure to have a redirect in there. So this website, the beta website, is where we have all the latest and greatest about Vitess. These different examples that I showed you earlier. There's documentation for how to set up Vitess from a developer's perspective. We don't have a lot of documentation right now for sort of an end user, but since there are so many different ways how Vitess can be deployed in different data portals or other settings, we haven't quite figured out how to best document it. But if you're a developer or a data analyst, you'll find a lot of very useful information on our website. So both sort of if you're a web developer building applications, or if you're a data analyst writing Python code to analyze single cell or spatial single cell data sets, you will find information on there, lots of tutorials, demos, etc. And just to give you a bit more of a flavor of how we're using Vitess in the HubMap data portal, I'll show you a couple of slides about the data portal and how visualization is incorporated there, where our vision was, and so our vision, meaning here in this case, it's my team. We are building the data portal user interfaces. So the data portal in HubMap is an effort by 5 or 6 different PIs, with each with their own teams. And our responsibility is really to build sort of the front end, the interface that typical users will interact with. And our vision was to get people to the data and to touch the data, interact with the data in as few clicks as possible, right? And so that's why we really spend a lot of time and energy into building visualization tools and interfaces that make it really, really quick and easy to see the data. So there's no need to download any data in order to just take a look at it and see, hey, is this something I care about? Do I want to download this, etc. So in the HubMap data portal, there's a standard faceted browsing web interface, where you can select data by data type, organ, filter it down to whatever you're interested in. And then once you select the data set, in this case, it's an autofluorescence data set. As soon as you open this data set, you see a brief abstract and then you see a visualization of the data. So this is just an OMITIF file that sits on a web server that is part of the HubMap data portal, and you can view that. There's other information on there, such as provenance. How was this, or where did this come from? So you can see from which donor did this come, right? So you can see the donor. You can see there was an organ, so a left kidney of the donor. Then there was some fresh frozen tissue, and then there was a tissue section, and then there was this autofluorescence data set generated here, and that's what we're looking at right now. So you can see all of this in here. And then moving on, you have the metadata in there. You can also access the data directly from Globus, which is sort of the research data management system that's sitting behind the HubMap data portal. That's where the link is down here. But to come back to the visualization, right? So this is just a single image. It's autofluorescence, so there's not even that much exciting data there. These images are usually used in the context of this project as sort of a reference to help map other data sets on top of each other, or different data modalities on top of each other. But so what you can do in Vitesse or in the portal, you can go from sort of this smaller view to a larger full screen mode. Basically, you can change the colors to a dark background to make it easier on your eyes to look at these images with a dark background. And then you can zoom in and all the standard stuff. As I said, you can change the color mappings on the fly, and we can do this not only for autofluorescence or any kind of fluorescence data. We can do this for standard histology data. So these are PAS images, but any sort of H&E stained images can be visualized. We have examples for mass spec data. So this is another data set. So this is a MALDI imaging mass spectrometry data set, where the challenge is not so much in the size of the data. This is only about 2,500 by 1,300 pixels. But the challenge here is the number of different mass spectra that you can select. So here the data is already pre-binned, and there are only about, I don't know, typically like 40 or 50 different channels. But being able to quickly go through them and display them is sort of one of the strengths of Vitesse here, because again, you don't need to download anything. You can just do this directly in the browser, and you can look at your favorite mass spectrum if you're interested to see what it looks like in this tissue sample. Here's imaging mass cytometry in 2D. Earlier, what you saw, this rotating block of tissue, was a 3D imaging mass cytometry data set. So we can do this, of course. As I mentioned before, we can do codecs. So codecs, and in this example, we have actually a fairly advanced data analysis pipeline in the portal. So we do segmentation. That's another team, not my team, that's developing that. So there's image segmentation. You see, there were not a lot of cells detected in this view. But then we do quantifications for all of the 47 markers that were used in this experiment. And you can see there's this heat map view down here. And in fact, there's a clustering of the cells. No cell type assignments right now, but that's coming. But you can see there's clustering. The clustering correlates with the expression patterns for the different, or the quantifications for the different markers in this case. And so you can, look at the data directly in the portal. You can start asking questions right here without the need to do anything, on your end. Here's another example. So this is a simple, non-spatial data set. So this is a 10x genomics single cell RNA-seq data set, UMAP scatter plot, heat map view down here for, in this case, we do have about 20,000 or almost 22,000 different genes. Unfortunately, they were mapped to ensemble gene IDs rather than gene symbols, which makes things a little bit harder, but you can see the ones that are shown here in the heat map view at the bottom were identified as markers for different clusters that were identified in this analysis. And then we also have sort of a violin plot. It's kind of hard to see here in the center for the gene expression for this selected gene by selected clustering. And so the test can support many different clusterings. It doesn't have to be only one, but you can load, say in this case, it's this latent clustering, but you can load all sorts of different types of cell type assignments in there as well. Here we're mapping gene expression levels onto the UMAP view as well. So you can see that this cluster that we were looking at here, this blue one and the orange one, the green one correlate with the expression of this gene. All right, so at this point, I think we're coming to the end. I want to really acknowledge my team. Mark Keller is a PhD student in my lab. He's been leading the development of the test. Chuck McCallum, senior software developer, responsible for much of the data portal in HubMap. Elon Gold, also a software developer who has contributed much to both Viv and Vitesse. Trevor, as I mentioned before, developed the Viv image visualization library, and then a couple other people who contributed, including my colleague Peter Karchenko, who's quite well known, I think, in the single cell field, who's been a great collaborator in this effort as well. I mentioned I'm hiring postdocs. Here are the people who would be, or here are the grants that would be paying those postdocs, among others. So I want to thank the NIH Common Fund and the National Library of Medicine for providing funds for both HubMap work and trainees. And with that, I would be very happy to answer your questions. And again, the reminder, I am hiring.
Thank you, Nils. Now is the time for participants to ask questions. I think you need to raise hand or type in your question. Then Vanessa and I will be able to unmute you, I think. Maybe I will start while others are thinking about the question. Nils, can you talk about examples of publication support, given the data objects are so complex and people take one image in the paper? What are the rules coming up for sharing?
So, so for publication support, I mean, I think if I understood your question correctly, right, I think there are two issues, right? One is, I mean, if you have just a single image as something you want to share, as I said, if it's a standard format such as OMI-TIFF or OMI-NGFF, you can store that, put it on a web server, and then you could include a Vitesse visualization to point to that, to view it. I think the problem that is not really solved by that is the fact that this image needs to live somewhere potentially in the long term. Yeah. And so there are not a lot of places really for imaging data. I mean, there's the image data resource, at the European Bioinformatics Institute, which is one place where people deposit their imaging data sets. But I am not even sure, if it's an expectation yet in the field to provide your images, or if people are happy to just take your publication and trust that you did the segmentation correctly and that you quantified everything correctly. So this is, I think, an ongoing question where I think no final word has been spoken. I think we would like to see more public imaging data. But even if you can get it stored somewhere, right? For viewing images like this on the web, there's a cost. Like in particular, if it's a commercial cloud, any data transferred out of a S3 bucket or Google Cloud storage will cost someone money. And how to do this in the long run is anything but clear. HubMap has a so-called hybrid cloud where some of the computing is in a commercial cloud, but the data is stored at the Pittsburgh Supercomputing Center. So we don't have to worry too much about the data access costs or egress costs, but it's certainly something that is not fully resolved when it comes to imaging data. And imaging data is just going to get larger and larger anyway. So I think it'll take a little while for that to be resolved. But I presume in the end, it'll probably just go the way how it goes with other technologies, where at some point you will have established methods that people trust. And as you say, okay, I did this algorithm, ran this algorithm, and we got this segmentation. Everyone will still say that's good enough, and we'll trust. You can look at yourselves. But I think we are still a couple of years away from that point. So there will be an interim where we need a solution.
Yeah. So still remind the audience, you can raise your hand or type in your question. I'll let Vanessa to help me monitor those.
I do have to say, having come from an industry space, actually, data storage is one of the biggest challenges that we face right now today, whether it's single cell or just sequencing in general, imaging, anything. And we really kind of as a community need to come up with a definite resolution to that, because it's getting more and more cumbersome, especially for labs to be able to maintain that data. But that's, of course, where all the information is. So we don't want to lose it.
Yeah. Indeed, I agree. Luckily, my lab is luckily not in the business of generating data. We're just trying to help people interpret and look at their data. But we do get a lot of requests to collaborate with groups who are basically just drowning in data and don't know what to do about it. And so in that way, we often do end up getting involved in this data storage issue as well.
Yeah.
So one bonus here is, right, so I think if we can at least avoid sort of these problems with like duplicating the data. So if you have formats that serve both as a storage format, but also can be used for analysis. And I think ZAR is a good example for that, right? So people can use a ZAR store to do analysis on the high resolution data while also using that same image to view it, or the same data set to view the imaging data. That is one minor plus, right? I mean, at least you don't have to make 2 copies of this thing. And then, having cloud environments can be useful as well. But it's non-trivial to really figure this out.
So Nils, I want to ask about your platform now put a lot of the data transformation to the end, kind of front of the stage. Users can apply. So I can see kind of a 2-part question. One is the risk of people misapplying a lot of decisions, getting something they like, then stop there. The second is, are there quite computationally involved tasks that actually will be slow, such as decoding of this cyclic hybridization data?
So I think this is an excellent question. Let me maybe start with the second one, because I don't really have a great answer for that one. Well, I mean, I can answer it. What aspects are slow? How do you deal with this in an interactive exploration setting? There are many analysis steps, of course, that are rather slow. I know that some of these codex images that HubMap is processing, take a very, very long time. I mean, it can take days to process fully, and at least with the pipeline that HubMap has set up for that. So of course, you cannot do interactive analysis on anything like that. So what we're doing is we're defining different sort of points along this analysis process at which we're grabbing the data or saving the results, and then we're bringing all of this together in Vitesse. And so you can look at the original input data set, the image, and you can look at the cell segmentations, which are sort of the first step, and then you get the quantifications, which are the next step, and then you get the clustering results, which is yet another step down the pipe. And then in Vitesse you can bring all of this together. Now what you can't do is through Vitesse you cannot modify the analysis, right? You cannot say, oh, I want to rerun this with different parameters. This would require someone to build a different analysis environment, which certainly would be possible. And we anticipate building this for some kind of interactive analysis steps in the future of HubMap, is the ability to relaunch an analysis with different parameters. But yeah, I think we're really relying a lot on pre-generated results when we're looking at analyses or data from these kind of data sets. So this is not something that you can do directly in Vitess, because it doesn't do any computation. Really, it relies on other analysis pipelines, or in the case of when you integrate into Jupyter Notebook or in R, then you use these environments to do your computation, and sometimes you just have to wait before you get more data to look at. But to answer your first question, I saved that for last because it's sort of a general problem in interactive data exploration, right? So how do you avoid people from sort of making mistakes when they, or sort of like introducing their own biases when they do data analysis or data exploration to generate hypotheses, right? This is really what this is about. The idea of interactive data exploration as a way of generating new hypotheses is probably how people use it most of the time. In some cases, people might use a tool like Vitess to confirm a hypothesis. They just want to, check whether things make sense. But most often, people try to find patterns and then try to explain them. And that is something that is common across all tools, no matter what kind of data you have. But whenever you do this sort of exploration and sort of information foraging, where people sort of go from patch to patch where there is interesting stuff, they might skip something. They might focus just on one particular aspect. And there are very few ways of preventing people from doing that. But one way of putting a little bit of a handle on that is to, and I'm now going to say something that's completely something we haven't been able to do yet with Vitess, but something that we're planning to do is to bring in provenance tracking. So they're sort of the idea of provenance tracking for like analysis pipelines is pretty well established in bioinformatics. But once it comes to interactive tools, the idea of sort of analysis provenance or visualization provenance has not, I mean, it exists as something my group has developed some approaches for in the last couple of years, but it's not sort of something that we do as a routine to build this into visual exploration tools. But we can track what people are doing, what people are looking at, and we can use that information, in fact, to for example, tell people, hey, you've only been looking at 1 out of 10 different genes. What's going on? So you can use this information if you can collect it to really understand user behavior. You can use it to guide them or correct them. I mean, correct is maybe the wrong term, but really to guide them to things they haven't looked at. And you can, of course, also replay an analysis, and you can go back and say, okay, you made a decision to explore this one set of cells when you were looking at the visualization, when it looked exactly like this. Why did you not look at this other cluster? So we can do these kind of things. And so there's a proposal that we recently gotten funded to apply Vitess to HubMap, to HTAN data, the Human Tumor Atlas Network. And for HTAN, we'll integrate the provenance tracking into Vitess so that we can capture these user interactions with the visualizations and therefore with the data, and then use that information to tell them that, what they've been looking at and what they've not been looking at, and hopefully address this issue that people just have biases and are not very comprehensive often in their explorations and miss interesting clues. So that's something that we will be doing in the future. And if you're one of those postdocs who hopefully will apply, it might be you who can work on this issue, which is a really interesting one.
Thank you for that answer. Yeah, I see Evan and Justin joined the panel. Do you or any other participants want to have another question? We're near the end of the hour. Well, hearing none, I want to, yeah, there are other words of gratitude coming in the chat. I want to thank you, Nils, for talking with us. We hope to have a chance to see you on other occasions. Yeah.
Thank you again for the invitation. It's been a pleasure to speak here today, and I'm looking forward to hearing from you, and we will probably cross paths again in the future. Take care, everyone. Yeah. Thank you. Bye-bye.